Hello students, have your seats. From last two sessions, we are continuously discussing regarding lysosomes. In first session, that was 15th session. During that, we have discussed regarding structure, origin and discoveries of the lysosome. In 16th session, we have gone through the various functions of the lysosome. Now we have to discuss the various type of lysosomes, various form of the lysosomes which are found in eukaryotic cells. With this session, we will conclude the discussion on lysosome. Let us start the types of lysosome. Actually, listen students, lysosome is a polymorphic cell organelles. Polymorphic cell organelles. You see this term, polymorphic. It made up of two part of word. Poly means many. Morphy pertains to the body structure, morphology. That means the lysosomes are found in many forms inside the cells. We will discuss here four forms. In those forms, they are found in eukaryotic cells. First form is primary lysosomes. Primary lysosome is the first form based on its origin. From there its journey starts. In first session, that was the 15th session, in that we have gone through the process of origin of the lysosome. We have seen, we have discussed how the lysosomes are originated by Golgi bodies with full coordination and association of a smooth endoplasmic reticulum. Again, I am repeating that process in brief. With the association of a smooth endoplasmic reticulum, the Golgi bodies, particularly the cisternae of Golgi bodies, synthesize the lysosome by the process of budding. Lysosomes are formed by the process of budding in the form of a small cyst-like structures, and then they are scattered in various parts of the cells. That time we discussed regarding GERL. GERL, the process for the synthesis of the lysosomes inside the eukaryotic cells. The GERL stands for Golgi associated endoplasmic reticulum lysosome synthesis. By this process, the lysosomes are synthesized. At that time, their forms are original. Their forms are primary. In that stage, they are called as primary lysosomes. Now we will discuss the second form. Second form is the secondary lysosome. As this name reveals that they are secondary in origin. That means after their synthesis, after their formation, the primary lysosomes gets the form of secondary lysosomes. Secondary lysosomes are also called as digestive vacuoles or heterophagosomes. You see this term, heterophagosomes. This term is made up of two parts. First, hetero. Hetero means another. Phagosomes means engulfing, intaking by a certain body. That body is lysosome. By this term, we are understanding here, it is quite clear, this form, secondary form of the lysosomes, engulf the certain material found in cytoplasm, and they produce digestive vacuoles. This serves as a digestive vacuoles to digest the engulfed material. So they are also called as digestive vacuoles. Now it is clear the secondary forms of the lysosomes is the secondary stage of the lysosomes after its origin. In that stage, they engulf certain cellular contents, microbes, and then they produce food vacuoles and digest those engulfed materials or microbes inside their body. Now we will discuss the residual lysosomes. You see this name, this term, residual. This part of name, of form, of the lysosome tells its own story. Residual means something has left behind certain process. Actually, this is the third stage. This stage, this form, comes after the second form. In second form, you have seen the secondary lysosomes or digestive vacuole or heterophagosomes engulf certain material. They serve as a food vacuoles. They digest those engulfed material. After digestion, certain materials are left undigested. That means after digestion, certain materials are residual inside the body of the lysosomes. So, this form of lysosomes is called as residual lysosome. Now we will discuss the fourth type of the lysosomes. That is the autophagic vacuole or lysosomes. During 16th session, we have already discussed regarding autolysis. Again, I am repeating that process. When the complete cell become weak or get worn out due to certain metabolic disturbances, due to certain other factors like aging. In that case, the lysosomes found inside that worn out cells digest the entire its own cell with the help of their digestive enzymes. This whole process is called as autolysis. That time we have discussed. In that stage, the form of lysosome is called as autophagic vacuoles or autophagic lysosomes. Now, here we have completed the description regarding various type of lysosomes, various forms of the lysosomes which may be found inside the eukaryotic cells. Now we will discuss two important terms which pertains to lysosomes. First is stabilizer. Inside the body, inside the cell of organisms, certain chemical substances are found. They stabilize the lysosomal membrane so that lysosomes cannot be disintegrated. Such type of chemical substance found inside the cells are called as stabilizer. Here we should know the certain examples of stabilizers which are found normally in the cells. First is cholesterol. Second is cortisone. Cholesterol and cortisone stabilize the membrane of lysosomes and protect the lysosomes from disintegration. Now, second term is levellizer. Levelizers are just opposite to the stabilizer. Those material, those chemical substances which are found inside the cells, which destabilize the membrane of lysosomes and cause the disintegration of lysosomes itself. Example of such substance are vitamin A, vitamin E, vitamin K. Apart from this type of vitamins, there are certain hormones like progesterone, testosterone. They also destabilize the membrane of lysosomes. And there are certain external factors like ultraviolet rays which also destabilize the membrane of lysosomes. Now, with this lecture, with this information, I am concluding the description or discussion on the lysosomes. In next session, we will start discussion on one of the very important cell organelle, mitochondria. Thank you very much.